Well, hello everyone, and welcome to the Research Bites webinar on nanobodies. Hello, Assistant Professor Helen Dooley. Hello, welcome. And hello, Associate Professor Gerald McInerney. Thank you very much for coming. I'm very excited to speak about this great topic. So first, I'm going to introduce myself. My name is Carmen. I am a field application scientist working for Thermo Fisher Scientific, and here in this Spanish Association of Researchers in Australia Pacific, I'm an industry liaison. We SRAP are a non-profit organization. We have around 300 members, and we have links with universities and research centers across this entire region. And the aim of SRAP is to disseminate quality science and facilitate collaboration opportunities to contribute to the cultural enrichment and knowledge advance of both Australian and Spanish and therefore European societies. We SRAP we get help and support from many different organizations like the Instituto Cervantes, the Australian Embassy in Spain, the Fundación Consejo España-Australia, the Fundación Ramón Areces. So thank you very much. Our main objectives are to help each other, of course, to connect researchers in the industry in Australia Pacific region, to help Australian research institutes and university understand the Spanish university system, and to participate in and influence the Spanish research system via a network of international associations. And in the long term, we want to be a part of changing the Spanish research system. This is the agenda for the webinar of today. I'm very excited because we have two great talks. The Assistant Professor Helen Dooley will start, and the topic is how sharks are helping us to attack disease. And then the Associate Professor Gerald McInerney will follow her with a topic that it's about the topic is an alpaca nanobody neutralizes SARS-CoV-2 by blocking receptor interaction. And then we will have a question and answer section, and then we will have a short scientific system discussion. Please ask your questions in the question and answer section of Zoom. You can write the questions during the talks, but we will respond them at the end in the Q&A section. And please make sure that you write the initial of one of the two speakers, either H or G. And yeah, this event is going to be recorded. So for those who couldn't assist, we will share it in the social media platforms. So stay tuned. And well, don't miss our next events. The next one coming is the Australia-Spain Research Forum that is happening in Sydney here. So we will be in the Macquarie University, but it's going to be transmitted online as well. And we will keep going with different Research Bites events. And yeah, we also have the Scientific Awards Premio Malaspina. So Helen, I'm going to, you can prepare your slide now. So let me stop sharing. So I'm going to give pass to Helen. So let me introduce and tell you about a little bit about Helen. So Helen is is an assistant professor at the University of Maryland, Maryland's School of Medicine in Baltimore in the United States, where her team studies the immune system of sharks. Their work aims to discover how these amazing animals protect themselves from infection and how this compares with humans. Much of their work to date has focused upon antibody responses following immunization, with particular focus upon a novel isotype that is found only in sharks. Helen's work has been used to create highly specific reagents for disease diagnosis and potentially treatment in humans and other animals. Please, Helen, proceed.
So first of all, thank you, Carmen, very much for inviting me to talk today, and good morning to everybody from a very rainy and windy Baltimore.
Well, thank you. Thanks to you.
Oh, pleasure. So today I'm going to be talking about our model system that we use in the lab. So we very interesting model system, it's not the usual, and how we're using this to help fight human disease. So I slipped this slide in just because I'm always super curious about how people got to where they got to. And when I tell people that I work on sharks, they immediately ask me if I'm a marine biologist. And that's not true at all. And I kind of got into this through the back door. And my PhD was actually in antibody engineering. So way back before antibodies were cool. I did my PhD in antibody engineering, where I worked on mouse antibodies, and actually was one of the first people to start working on camel antibodies. That didn't go according to plan, and I can tell you all about that at some later stage, but it didn't go according to plan. And during my PhD, I became excited about, kind of antibodies in non-traditional models, and that's how I got into shark research. So after my PhD, I jumped over to UMB, where I became a postdoc fellow and really started learning about comparative immunology, where we look at different immune systems across different animals and compare them. I'm still working in a shark model. And I was there for a few years until I was headhunted by Wyeth and moved into their system for a few years through the transition with Pfizer doing biotherapeutic development. So after Pfizer, I actually decided to move back into academia, kind of take back control of my research, and got offered a position as lecturer at University of Aberdeen. And this is one of those things that, I talk to a lot of PhD students and postdocs about, because there seems to be this thought that it's really difficult to make the transition from academia to industry or from industry back to academia. And that's certainly not my experience. You build skills in both of these situations, and certainly in the transition from Pfizer back into academia, one of the things that really appealed to the people that I interviewed with was the fact that I had worked in industry and I had this experience that I could share with students and with other people, and kind of a little bit of inside knowledge. And it also helped me in recent years. I've been now in Baltimore for 4 years. I was offered a position as assistant professor here in the medical school, but based in the Institute of Marine and Environmental Technology. So again, they were really keen that I had this industry experience. And this is our building up here. So on a beautiful day, we're based right on the waterfront with the Coast Guard cutter Taney parked outside our building. So we're well defended should anything happen. And the reason I'm here is obviously, my model system, we don't have mice, so we don't need a mouse room or anything, but we do have sharks, so we need an aquarium, and we're based on the waterfront. We have a working aquarium in the basement of our building where we keep our animals, and we can go down and we do what we need to do. So really what my lab is focused on is how the vertebrate immune system has changed over evolutionary time. So we're really interested to see, the way my students put it is that if we only look at humans and mice, essentially we're only watching the last episode of Game of Thrones, or we're only reading the last Harry Potter book. You know what happened, but you have no idea how you got to that point. So we kind of start at the beginning of that story with the earliest vertebrates, jawed vertebrates that have a mammalian-like immune system. We start with sharks, and then we work our way through and we look at all the changes that have happened to the system over evolutionary time to kind of get the story of these molecules and how they got to where they got to and how the system got integrated the way it did. And what we're hoping is that that will help us understand human diseases like rheumatoid arthritis, where things go wrong. You know, so if we can understand the system a bit better, perhaps we can figure out ways that it goes wrong and come up with better answers. And this is really what I'm going to focus on today, because when we were doing our work, we actually came across this molecule that we thought could be applied to this goal of being able to diagnose, prevent, or cure disease in humans. And we're not biased. We will also look at other animals as well. So, you know, we talk about human and non-human animals in my lab. So everybody always asks me why we work on sharks for our research. And when I talk about sharks, basically I'm lumping everything together in the cartilaginous fish category. So you guys will be aware of, you know, sharks and skates and rays, maybe not so much the chimera, which are the kind of most ancient cartilaginous fish lineage, and these like little weirdo guys down here at the bottom. Oh, sorry, let me just pull up a pointer. Like these real weird deep sea guys down at the bottom, they're really evolutionary ancient lineage. So we work on sharks, like I said, because they actually have a mammalian-like immune system, but they emerged about 500 million years ago. So they're very ancient lineage. They've been through a load of bottlenecks, like 5, depending on how you count them, about 5 mass extinction events. We think there's about 1,200 living species at the moment. And just to put this into some sort of context, we as a species, Homo sapiens, have only been around about 0.3 million years. So we're dealing with these animals that have been around for a very long time. And I always like to stick this in because I think of dinosaurs as being ages and ages and ages ago, but sharks outlived them by about 250 million years. So they were there around a long time before the dinosaurs, and they're still trucking on today. So despite this, they actually have everything a scientist dreams of, if you're me, which means they have polymorphic and polygenic MHC. So if ever I wanted to do a tissue transplant on a shark for some reason, I would have to go in and tissue type it and match the tissues. And this basically enables them to respond to a huge range of different pathogens that they experience as a population. Just like you and I, they also have T cells with somatically rearranging T cell receptors. And most importantly, for what we're going to talk about today, they also have B cells that somatically rearrange Ig genes to enable them to secrete antibody for protection. And most of you will be familiar, if you're a human, that you have 5 different types of antibody, so IgM, D, G, E, and A. And when we look at the shark system, and all of those antibodies are what we in the nanobody trade call conventional antibodies, that are made up of 2 heavy chains. So the blue chains are the heavy chains, and 2 light chains joined by this highly flexible hinge region. And if we clip those apart, basically we get these antigen binding regions that are able to bind to antigen but without Fc function. And for the longest time, in fact, when I was back doing my PhD, what we were trying to do was take these, mouse monoclonals or human antibodies and basically break them down to the smallest constituents that we could that still bound to their antigen. So a lot of work happened on these FAB fragments, or just these 2 domains up at the top, FC fragments, or usually linked together by some sort of linker or disulfide bond, these single chain FV fragments. And the whole idea of this was to get them smaller so that we could make them in bacteria more easily and cheaply, and be able to use them in different, like I was particularly looking at them for detecting pollutants in the environment. So we were trying to get them small. The problem with that is then you lose all the stabilizing forces. So I spent much of my first part of my PhD sat in a cold room for 12 hours a day trying to purify these things and do ELISAs with them. Because yes, they were small, and yes, they bound to their target, but the minute you heated them up to anything above 25 degrees, they would just fall apart and be completely useless. So obviously that isn't ideal if you're going to try and use them in any sort of sensible application. You can't have something where it has to be, or you can have things that need to be refrigerated, but it gets complicated and it gets expensive. And your PhD students certainly don't thank you coming out of the cold room looking like an Eskimo bundled up in their jackets. So kind of during the time of my PhD, we were looking for better solutions to this. I did a little bit of work on the camel antibodies, but like I said, I got really interested in the shark system fast. And the reason for that was because it had been known for a long time that sharks had an IgM molecule, which is very similar to the IgM molecule that's found in us. They also have this isotype called IgW, which is essentially the same as IgD that we have. But as well as those two isotypes, they had this completely novel isotype, which is called IgNAR for new antigen receptor. And new antigen receptor, because basically when it was found, we weren't actually 100% sure that it was an antibody and that it was doing kind of antibody humoral type protection. So IgNAR is interesting because you can see from the little diagram, it doesn't have light chains. So when it was originally found by Andrew, who's in Dr. Martin Flanagan's lab, who I eventually postdoced with, they did EM of these molecules, and you can see basically you have the little constant domains down here, and then the variable domains that stick out either end, like little ears on a bunny. And it's these that actually do the antigen binding. So these guys in red on this molecule, and then all the constant domains here. So while the whole Ig molecule is actually much bigger than kind of a human IgM or IgG, the actual binding regions are much smaller. And because they're only single domain, we get around this problem of having to keep two things together. And we thought that this would give us extra stability and enable us to produce them more easily. So lots of different animals we're going to hear, and I'm excited for the next talk. We're going to hear all about camels and llamas and alpacas and vicuñas, all have these special single domain antibodies that don't need light chains to be able to bind to targets. But we've mainly been focusing on nurse sharks for about the last 20 years now. And people often think that the two things are very similar, but in fact, so the camelid antibodies are actually derived from a normal IgG. So it's, it was found out a long time ago that they had an IgG1 that managed to lose this CH1 domain, and then you had mutations that solubilized the interface region to give you the special VHH antibodies, and that these could be kind of cloned out of an immunized animal and give you the 15 kilodalton VHH nanobodies. So the shark system is completely different to that. So these have evolved through convergent evolution. So you're getting the same thing, but by completely different pathways. And in this case, what we think has happened is actually a TCR variable region from this special molecule called NAR-TCR that, again, only sharks have, has actually invaded a cluster for IgW. And you've kind of got a soluble TCR region on the stalk of an Ig, which give these guys a slightly different structure to the normal camelid VHH antibodies. So what do? We figured out that if we immunize a shark in the correct way, and that took me about 3 years to figure out, and then we take blood samples, what we see is an increase in the antigen-specific IgNAR levels over time. So if we immunize our animal correctly, just like you and I, we get an increase in the levels of this antibody able to bind to our antigen. So in this case, first time we tried it, we just went in with lysozyme. It's a good model system. And we're seeing this really nice increase. And this was very specific to the antigen that we were giving the animals. So we finally got figured out, how you would immunize a shark. When I talk about immunizing sharks, I think people think that this is what I actually do. So for all the reasons that you can think of, this is not a sensible idea. First of all, great whites are big and bitey. I would love to work with great whites, but they're not the most tolerant of animals, I guess. Also, you think about it in terms of just the logistics of it. We can't keep animals this big in cages in tank systems very well. It doesn't do them any good. if we're going to immunize animals, we want them to be healthy, we want them to be happy. Also, you know, if we were out at sea, you immunize an animal, then you let it go, how do you ever find it again? At the risk of losing an arm to take a blood sample. So instead we work with, animals like this. This is a cat shark feeding frenzy. So much easier to handle, much easier to keep, much easier to feed. This system and our nurse shark system, you know, we've been working with for over 20 years, so we have all the tools that we need to be able to do this. It's not like working with mice and humans. We can't just go to Sigma and buy antibodies, so we have to make all the tools that we need. And we've built those up slowly over time. So we tend to work with these systems where we can actually keep animals happy and healthy, immunize them easily, and go in and take blood samples when we need it. So what we do now to generate our libraries, we generally use nurse sharks, and we will go in, we'll immunize our shark with the desired target. So just due to, you know, body temperatures and things like that, this takes a bit longer than in humans. We're normally looking at 3 to 4 months before we get really high antigen specific IgNAR titers. But at that point, we can take a blood sample from that animal, isolate the RNA from the PBLs, and then using literally 2 primers, we can amplify up the entire VNAR repertoire and clone that into a phage display vector. And anybody that's worked with, you know, I've worked with human and mouse libraries in the past, and you're dealing with, you know, literally 20 and 30 different primer sets. This is a huge boon. We can basically now build a library in, you know, a day and a half or 2 days and get a 10 to the 10 library at the end of it. So we have our entire VNAR repertoire in our phage display vector, and we put that into bacteria, infect it with helperphage, so that we get our VNAR domains displayed on the surface of the phage, which means then we can come in and pan that against an immobilized antigen. So whatever we immunized our shark with, we're able to pull out the VNARs that are specific to that. And here, because we have a link between actual function and the, you know, plasmid that are inside the bacteriophage, we're able to take those plasmids, clone them into bacteria or, you know, yeast or mammalian cells, whichever expression system we like, produce these recombinant proteins, purify them, and actually then confirm binding and test in whatever downstream application that we're interested in. So this is kind of our system that we go through. The end result of that is that we get these, this is a crystal structure, a ribbon of a crystal structure that we made. We get these extremely small, so about 12 kilodaltons, highly stable binding regions that are able to bind our target with very high affinity and specificity. So we have antigen binding domains that can literally discriminate proteins that are, you know, over 98% similar. Dependent on where they bind. The beauty of these things is because of the evolutionary distance between sharks and humans, we have a lot of success raising the antibodies against so-called hard-to-hit targets. So you can imagine if you have a situation where you have a protein that's involved in human oncogenesis, but you put that into a mouse, and because the protein is 99% identical in a mouse, that you don't get a good antibody response and you can't raise antibodies. In that case, if we look in sharks, generally the identity between those 2 proteins will be less than 70%, just because those animals are separated by 500 million years. So that means that the shark immune system recognizes it as foreign, and we're much more likely to get these high affinity antibodies against these hard to hit targets. And we've worked with a couple of groups that have had real trouble getting antibodies against their favorite target to raise agents for them that they can use in their research. Also because they're small, and people have seen this also with the camelid antibodies, is because they're small, they're able to fit into spaces that conventional antibodies can't bind to. So in this case, what we have is, in this antibody, the CDR3, which is shown in yellow, so the main binding determinant of this antibody, is actually inserting itself into the active site of the enzyme, and is able to inhibit enzyme function. And then, also the beauty of working with single domains is that they're super easy to engineer. So once you have them, you can put them onto, if you're interested in working in mice, you can put them onto a mouse FC. You can put whatever tag you want onto them. You can string them into, strings like a bead. You can form all sorts of, whatever novel configuration that you want to come up with. Because they're single domains, you don't have to worry about keeping them in contact with the partner. So we're able to engineer them into lots of different formats dependent on the use that we want. So like I said, first antibody that we got was kind of a surprise. We got the crystal structure, realized that it bound straight into the enzyme active site. And you can see here, so this is our, we actually did a study to show that, if we have a control VNAR or no antibody there, you get one rate of enzyme activity. But if we added our anti-Hel VNAR, that you actually slow the rate of activity. So this is actually inhibiting the enzyme function. Again, when we got the crystal structure, it became apparent why. This is the oligosaccharide, so the normal target of this enzyme that binds into this active site. And you can see here, this CDR is literally mimicking the oligosaccharide, the normal target, and binding itself into the active site. There's been lots of kind of mouse antibodies raised against lysozyme in the past, and not a single one of them binds into this epitope. So they all tend to bind to these flat surfaces on the surface of the molecule. And this is kind of one of the most famous ones, binding up at this top, but none of them are able to bind here. And I think some of the camelid antibodies bind into this site as well. So when rapidly building up structures for these things, and all of them are shown here with different targets. So the targets are in gold, and the antibodies are all labeled up in exactly the same way. Here, the CDR3, the main determinant of binding, is in red. And we can see, these are the original ones that I did. So we have the CDR in red, and this is CDR1 at the back. So this is using 2 loops of the antibody to bind. In this case, we actually have 3 loops up at the top of the molecule that are binding. But then in this case, the antibody is using its CDR3. And actually this strange little loop I talked about, how these are really T-cell variable regions, not Ig variable regions. So they don't have a CDR2. Instead, they have this little loop that's like a belt that wraps around the outside of the molecule. But this is also used in antigen binding. So these molecules can use this top surface, but they can also use the side surface, which gives them lots of different ways that they can interact with different targets. And it's probably why we're getting such nice high affinities against different things. If this doesn't fit, then they use the long side. And again here, we're seeing CDR3 and this hypervariable loop HV2 that are doing all the binding. So have we been applying these? So a lot of the work that we do is working with other teams. So we raise antibodies. People come to us and they say, we have, we're having problems getting antibodies against X, Y, or Z. Can you help? So we do a lot of collaborative work, which means that some of it I can't actually talk about. But one of the things that we did that was really successful and has been published, so I can talk about it, is using these as better diagnostics for disease agents. And this kind of came out of it was not the last Ebola outbreak, but the one before, I think 2014. Where there was a huge Ebola outbreak, and what was happening was people were sending test kits out to where they needed to be, and because cold chain couldn't be maintained, by the time the kits got there, they couldn't be confident that they were actually still viable or not. And this was perceived as a huge problem. So we were approached by both the UK and the US defense agencies to see if we could maybe raise antibodies that were more stable against target that could be used in these assays instead of the traditional antibodies that they were using. So in a feat of paperwork magic, never, ever, ever suggest this to your health and safety committee. We got permission to immunize some of our sharks with inactivated Ebola virus. So it made for some interesting reading in terms of EHS. But what we found is after a couple of shots of inactivated virus was that the animals, we actually immunized with Ebola Zaire. And the animals responded really well to the virus. And we're finding with our sharks is that viral targets generally work really well. So it seems to be something their immune system is set up to respond to. But there was also cross-reactivity in terms of the response to other Ebola virididae. So this was really interesting because if we could get something that actually could bind to lots of different Ebola virididae, then, you know, we would have this kind of really quick and easy diagnostic that would pick up any sort of outbreaks that were out there. So we immunized our sharks, we built our libraries. At the same time, our group in the UK did exactly the same thing with mice. So the idea was that we were going to do side-by-side comparison of mouse antibodies versus our shark signal domains. And this was the lead clone that came out of the mice, and what they were interested in was temperature stability. So the way we assayed that was to, you know, we coated an ELISA plate, then we took the antibodies and we heated them for 3 hours at different temperatures. And then at the end of that 3-hour period, added to them to the ELISA plate and looked at residual binding compared to a standard that had just been kept in the fridge. So in this case, you know, the mouse antibodies do great at 40 degrees, 50 degrees. Surprisingly, we're doing okay at 60 degrees, but by 70 degrees had kind of completely lost any binding. So, you know, this, like I said, they behave better than we thought, but, you know, 70 degrees, so a hot day on the back of a truck would basically kill these antibodies and prevent them from functioning. If we did the same assay with our lead clone that came out of the sharks, you know, we're doing great at 40, 50, 60, 70, even at 80 degrees, we've still got almost 50% of binding. So we kind of managed to shift the temperature. I think even like 100 degrees, we were still getting 20% activity after 3 hours, you know, after boiling them for 3 hours. So in terms of being able to shift the stability of this assay, you know, these clones would take us up an extra at least 20 degrees. That doesn't sound so dramatic, except what it is the difference between having to keep these things, you know, you make your kit, having to ship it out to, you know, wherever it goes in refrigerated containers on the back of a you know, refrigerated truck. When you get to the other end, you have to have power to keep refrigerators going, and you know, you can never be 100%. If that doesn't work, you can never be 100% sure that your assay is actually working properly. Whereas in this case, you might be able to take that same assay and stick it in the cool box on the back of a bike and get it to where it's needed more efficiently and cheaply and quickly. So, like I said, we're working on lots of different projects. We're looking at the potential of these things as diagnostic agents and therapeutic agents. A lot of our work is for, like I said, these hard to hit targets that are involved in human cancer. That is not because sharks do not get cancer. I know that I get asked this all the time. Sharks absolutely do get cancer. You know, and there's documented evidence of that. It's just this evolutionary distance, like I said, between the human targets and the shark targets. We're doing a lot of work on viruses lately. You know, we think these things have potential for toxin neutralization. We're talking to a few teams about projects that involve that. But basically, I think, you know, it's literally up to the imagination of the researchers that are working on them as to what we could do with them, because they're so easy to manipulate and to get. And I'm certainly not, I'm not of the belief that one format is better than another. So, you know, we never should work on human antibodies or mouse antibodies or camel antibodies, and shark antibodies are the answer to everything. It's my belief that, you know, all of these things are tools in our toolbox, and the more tools we have, the better chance we have of picking the right tool for the job. So in this case, you know, I feel like maybe, you know, viruses or these hard to hit targets is when you would choose the shark system. For other things, you might want a camel antibody. For other things, again, neither of those would be great, and you would want, you know, a human antibody or a mouse antibody to do the job. But it just gives another option to be able to address some of these problems. And actually, we were lucky enough to be, you know, anybody that's interested in reading more about this, included in this special article that was produced in Science in May 2018 that we're talking about, you know, these single domain antibody formats. And they were very excited. This was actually me bleeding a shark. They were very excited to come out and visit our sharks. Nobody comes to see me. Everybody comes to visit my sharks. But we did get lots of beautiful pictures in the article, and they talk a lot about the different work that's going on. Okay, and then, you know, really I just sit in my office and drink coffee, and everybody else does the hard work. So Hanover, my PhD student, Dinesh, they've both been working on IgNARs, and our collaborators in DSTL and USAMRIID that we work with on the Ebola virus project. And then obviously, let's see, these guys that are the stars of the show. So these are on our sharks, hanging out. And they all think I've got food, so they're going to start coming up. And that is it from me.
Well, thank you very much, Helen. That was a fascinating research. Thank you very much for sharing and for presenting your work. So now we are going to give pass to our second speaker. And then after that, we will pass to the round of questions if you would like to ask any questions to our speakers. So Gerald is our second speaker. Gerald McInerney is an associate professor of virology at the Karolinska Institutet. His research group have a long-standing interest in the molecular and cell biology of viral infection. They have studied how cellular metabolic and signaling pathways are modulated by infection. More recently, they have established a nanobody identification platform and have generated specific nanobodies against numerous viral targets. Since early 2020, they have been focused on developing nanobodies against SARS-CoV-2 with diagnostic and therapeutic potential. So I'm very excited to hear your talk, Gerald, please.
Thank you. Thank you, Carmen, for the invitation. Can you hear me and everything? And see my screen?
Yes, perfect. Mm-hmm.
Okay, so thank you for the invitation. It's a pleasure. It's a story that I'm going to tell you that we're very proud of in the lab, so we're always happy to talk about it. And thank you very much to Helen for that fantastic talk with great introduction to the single-chain antibodies. I'm something of an imposter in this world because I just became interested in these as molecular tools just a few years ago. And the story I'm going to tell today is really our first publication in that area. I'm primarily, I don't have a slide to explain my background, but I'm a virologist, as you said. I've studied virus disease, molecular biology of virus infection since all the way back to my PhD, and I've been at the Karolinska Institute in Sweden for 20 years now, first as a postdoc and now as a group leader and associate professor. So I'll just get straight into the story. So that sort of background sort of explains why we were in the position to be able to react quite fast to the SARS-CoV-2 outbreak as it was then and pandemic as it is now. So I'll tell you about that, and you'll get a sense of how we were positioned to be able to do the work that we did in quite a rapid manner, in quite heavy competition from around the world. But we'll get to that. So this is kind of the artist's impression of the SARS-CoV-2 particle. You'll have seen this in many different forms with different colors, of course, always fake colors. But so this is what the virus particle roughly looks like. And in the blue, light blue here is the spike glycoprotein trimer. So this is a trimer, so it's 3 molecules that come together on the surface of the virus particle. And this is the protein that is critical for perhaps the most important step of the life cycle of the virus, which is the entry, selection of the host cell and then entry into that cell. So this protein is responsible for binding to the receptor. So it has a domain known as receptor binding domain. And the receptor is angiotensin 2, angiotensin converting enzyme 2, so ACE2. And the protein also directs fusion with the host cell membrane. So after the virus finds its chosen cell, this protein is also involved in the fusion and entry of the virus. So it's a critical protein for the virus and a very important target for neutralizing antibodies, not just by the immune system in infected individuals, but also labs like mine that are trying to develop antibodies and drugs and other molecules that target this step. So the virus, as you know, was kind of emerged towards the end of last year and was finally isolated and described, and the genome of the virus was published on the 10th, I believe, of January 2020. And labs around the world reacted extremely rapidly to that, and one of those labs was a lab professor by the name of Jason McClellan in Texas. And his team reacted extremely rapidly and cloned the spike protein. So this light blue protein that I showed in the previous slide, cloned it so that they could express it and solve the structure by cryo-electron microscopy. And what this slide here is showing is the solution to that structure. So this is the domain structure of the protein, the N-terminal domain. This is the receptor binding domain that binds to the receptor. This is a critical for that reaction, and I'll talk a lot about this domain. And then there's an S1, S2, so the spike 1 and spike 2. There's a cleavage point here, which occurs during the entry process. And so when the RBD engages with the ACE2 molecule on the surface of the host cells, the S1 portion of the protein is lost, shed, so it's cleaved and then shed from the particle, and the S2 portion, which is here connected to the viral membrane by this transmembrane region, undergoes a conformational change that leads to the presentation of this peptide, the fusion peptide, into the host cell membrane and fusion of the virus and host cell membrane and entry then of the genome into the cell. And so what Daniel RAAP and colleagues did was to make some mutations in the S2 domain, specifically here in the central helix, such that this conformational change can't occur. And they were able then to solve the structure by cryo-EM of what we call the pre-fusion stabilized conformation of the spike protein. So this protein is not able to undergo those conformational changes, and makes it possible to, or made it possible to solve the structure. And the structure is shown here, and I'll be talking a little bit about this complex, so it's worth taking a minute to look at it. It's a trimer, and the trimers are the three monomers are shown in white, in gray, and then in this colorful ribbon diagram. And you can see from the colorful ribbon one that the RBD, which is the receptor binding domain, is here at the top of the complex, and the N-terminal domain is here on the side, and the other domains. And you can see the fusion peptide here, which is this light blue or cyan colored peptide here, which is involved in the fusion. And what you can also see from this is that the other two, so the white and the gray monomers in the complex, have the RBD domain in the down position. So it's kind of hidden. This is referred to as the 1 up 2 down conformation. So one of the RBDs is in the up conformation, and that's the one that will engage with the receptor. The other two are in the down conformation, and it's thought to be a mechanism to evade binding by conventional antibodies. Okay, so as I said, this was an extremely promising target for neutralizing antibodies, and this paper will I'm sure go down in the history of the COVID-19 pandemic as one of the landmark publications. And we were very inspired by this and thought that we could make neutralizing antibodies to this complex. And our particular strategy was to use single chain antibodies from camelids, also present in sharks, as Helen was saying, but we've been working for a few years in my lab on single chain antibodies from alpacas. So all of the animals in this family, so including camels and llamas and some of these other wonderful smaller creatures, are able to produce single chain antibodies, just like Helen was saying. So again, I show this picture of what we call conventional antibodies that have this heavy and light chain, two different genes, and then they form dimers, so that the heavy and the light chains are roughly in this kind of conformation. But the camelids, at some point in their evolution, have lost from one of their, or some of their heavy chain genes have lost this constant region, this one here, so that they don't have interaction with the light chain, and the antibody is then expressed as a single chain. And what's interesting and important is that they still maintain antigen specificity and affinity to some degree through the single variable chain. And so they can be more easily cloned just without having to perform single cell sorting. And then in the lab, we can clone these single chain antibodies and make them even smaller to these nanobodies, also called VHH, which are very similar to what Helen was describing for the sharks. And they're about 15 kilodaltons, so approximately 1/10 the size of the conventional antibodies. And they can be manipulated and used much more easily in molecular biology labs than conventional antibodies. So, for example, some of the things that we can do, and we have been doing with nanobodies, is we can perform direct conjugation with fluorophores, so we can use nanobodies for microscopy studies, and because they're smaller, you get better resolution in some microscopy applications, and we're kind of attracted to using them for that idea. There's a number of other things we can do. I don't need to go through all of this list, but one can make dimers, either homo or heterodimers, or even tetramers, as I'll show. So you can link nanobodies together to make higher antibodies with higher avidity, or multiple epitopes that they target. So for various reasons, one would want to do that. And some of another reason why we were attracted to use these molecules in the first place is that they have been shown to be very useful as crystallization chaperones. So for proteins that are maybe flexible, or for other reasons are difficult to solve structure by crystallography or by cryo electron microscopy, had been shown by co-expressing them with specific nanobodies would serve to stabilize the proteins and make them more easy to solve. And we were sort of attracted to this application of nanobodies, but also this a few years ago. So as I said, my interest just started just a few years ago, and that was when this guy, Leo Hanke, wrote to me and suggested that he come and join my lab. He was a PhD student at MIT in Boston in the lab of Hidde Pluk, who's been a very successful user of the nanobody technology for making nanobodies against viral proteins and studying them. And Leo was a sort of an important member of that team when he did his PhD. And then he came to my lab and proposed that we make panels of alpaca-derived nanobodies against the non-structural proteins of chikungunya virus. So I didn't talk about that, but our original interest, or my background interest before SARS-CoV-2 came along, was chikungunya virus, which is a alpha virus, a mosquito-borne virus causing fevers and diseases in the tropical region, similar to dengue fever. And we've been studying this virus for a long time, and we thought it would be very interesting to have nanobodies against all of the non-structural and also the structural proteins. And so Leo came and he set up a workflow similar to what we heard about in the previous talk. So we immunize alpacas with various proteins that we produce in the lab. We clone them into phage display libraries, exactly like Helen was saying, and pan them on immobilized antigens, and then we can select for the better binders and better expressors and various other properties that you would want your nanobodies to have. And Leo was getting to a very productive phase of that project. We have nanobodies now against the whole proteome of the virus, and we were wondering how we were going to start to prepare those projects for publication when when COVID started, and when the virus became obvious that this was going to be a major medical global health problem. And in conversation then with Ben Merle is an assistant professor of virology here at our department and a computational biologist. He has a lot of experience in developing antibodies against HIV, both from the practical and also the computational aspects of that. So Ben very convincingly argued that we should stop working on chikungunya virus in January this year and put all those nice projects on the shelf and jump on the idea of making nanobodies against SARS-CoV-2, and that's what we decided to do. Leo and Ben and I decided to go full speed ahead on that. And so just to tell you about some of the before I talk about the nanobody that we developed. So we contacted this lab that had solved the structure of the complex, Jason McClellan's lab, and they very generously shared with us, as they have with many other labs, the clone of their stabilized prefusion spike construct. And so we started to produce that and to use it in our experiments, but we knew from the start that this was going to take time to develop. And so we cloned ourselves the receptor binding domain, because we knew that that was going to be a powerful antigen. If we could have antibodies directed against the RBD, they would be very potently neutralizing against the virus. And we also used some soluble of this S1 subunit, the subunit that I told you is lost after receptor binding, but it contains the RBD. So these are the proteins we used initially. And this is our immunization regimen. So this is Tyson, who is an adult male alpaca, and this was the animal that we immunized. Tyson lives in a research farm in Germany, so we don't have the luxury of being able to go downstairs to our animal facility and work with these fluffy creatures. We send the proteins to a farm, so we've never actually met Tyson. I hope someday to go and visit, but Tyson was immunized with over a 60-day period with 2 immunizations with the S1 protein, and then he was boosted with this smaller RBD protein. And this was our attempt to develop nanobodies against the RBD subunit, or RBD, the receptor binding domain, which we hypothesized would be neutralizing if exposed to the virus. So towards the end of that 60 days, we received some serum from the animal, and we tested it just with a very simple Western blot, and we found that Tyson's serum reacts very strongly to the spike complex and the RBD proteins when just run on a conventional STS page gel. So that gave us some confidence that there was antibodies. Now, of course, these would be both conventional and single chain, but nevertheless, it's a good indication that there'll be some of the single chain antibodies present. And we also performed an experiment that I'll explain more in subsequent slides, but this is just seeing if the antibodies in Tyson's antibody repertoire are able to block infection with the virus, with SARS-CoV-2 virus. And we found that indeed we were able to detect some neutralization. It wasn't extremely high, but there is some activity there. And so that gave us confidence to go ahead, that there would be antibodies there that are blocking infection with SARS-CoV-2. So this was all very early. This was already back in March or April this year when we had these results. So we were very excited, and Leo and Ben and the other guys in the lab were working sort of day and night at this point. And so what we decided to do is to combine Ben's experience with next generation sequencing of antibody repertoires with Leo's experience of performing binding screens with the phage display. And we performed using the nanobody library that we developed from Tyson. We performed 2 rounds of phage display on immobilized RBD antigen, so looking for nanobodies that bind to the RBD. And after 2 rounds of phage display, at each, both at baseline and after each round, we were performing the next-gen sequencing. And we saw that one of the nanobody sequences, which we called Ty1, because it was the #1 antibody from Tyson, this sequence was enriched 10 to the 5-fold after 2 rounds of phage display. So that's an extremely strong enrichment. And we also knew from the binding screens that it was a very strong binder to the RBD. And we also knew from some other, we had ideas that it was also expressing very well in bacteria, and that it was behaving well in the way that one would, a protein biochemist would like a protein to behave. It was stable to temperature and that. So we knew we had a really good nanobody, and we decided so all of our downstream experiments were done with only Taiwan. And normally within our projects, we would maybe have taken the top 10 candidates here and gone forward with all of our analytical experiments with a panel of these. But because we wanted to be fast, we just took Taiwan and went ahead with it. And this is the sequence of the nanobody here, which we presented in our paper. And yeah, we just wanted to be completely open with what we had, since the funding we received was so-called public health emergency funding from the EU. So one of the stipulations is that we be completely open and publish all of our data within 30 days. So we decided to do that, and we published the sequence of the antibody. So anyone could essentially clone this nanobody if they wanted to. Okay, so one thing we wanted to do is see how specific it was and see if the Taiwan nanobody could be used as a tool for detection of the spike protein in the lab, or perhaps in diagnostic assays, which is something we've been following up on, although I'm not able to talk about it today since it's not published yet. But we were able to take Taiwan. This is supposed to be the Taiwan nanobody here, and we were able to directly conjugate it to a fluorophore, which is known as 635P, so it's red fluorescent. And then we're able to use that as a tool to stain infected cells. Okay, so we got SARS-CoV-2 virus from the first patient here in Sweden that was infected with the virus, and we isolated it and used it to infect cells. And then we stained those cells with an antibody that recognizes double-stranded RNA. So this is a conventional antibody that recognizes double-stranded RNA, which is only present in infected cells. So you don't have double-stranded RNA normally in non-infected or mock-infected cells. And the Taiwan nanobody, cut a long story short, it very brightly stains virus-infected cells and does not at all stain non-infected cells. So that tells us that it's somewhat specific, or perhaps very specific for the viral spike protein. We're also able to do flow cytometry. So these are 293T cells that are either transfected or not transfected with the spike construct. And we use the Taiwan 635P construct to stain those cells. And again, this tool has lent itself very well to flow cytometry application for detecting virus-infected cells. So again, very nice result that we're very happy with, but also I want to draw your attention to the background staining, which is really undetectable above the unstained cells. So we're really happy that this is a specific antibody that binds to the spike protein. But of course, what we're interested in is whether we had neutralizing activity, so whether the nanobody is able to block infection with SARS-CoV-2. And because we wanted to do a lot of experiments in this way, we decided to use a pseudotype pseudovirus assay. So this is not using the infectious SARS-CoV-2 virus, but rather using a lentivirus that expresses luciferase, which we were able to pseudotype with the spike protein from SARS-CoV-2. So this is not SARS-CoV-2, but it enters cells in a SARS-CoV-2 spike-dependent manner and expresses luciferase. And so that gives us a readout with a large linear range in which we can measure virus replication as a direct measure of virus entry into the cells. Okay, and so what we did was we pre-incubate the pseudovirus with the nanobody, and as we increase the concentration of the nanobody, we get a decrease in the luciferase signal when we use that then subsequently to infect cells. And so that tells us that this nanobody is blocking infection with SARS-CoV-2 virus, and notably it doesn't block infection with a virus that's pseudotyped with the VSVG protein, which is a very different virus protein, and an irrelevant nanobody is not able to block the SARS-CoV-2 pseudotyped lentivirus. So we were quite happy with this. So the IC50, in other words, at the point at which 50% inhibition is reached, is at 0.77 micrograms per mil, which is reasonably okay neutralization activity for this virus. But what we also did was to perform a trick where we expressed Taiwan in fusion with the human FC domain. So we're making now a molecule that looks more like a conventional, or looks more like a single chain antibody. So it's now present as this dimer, so it's now bivalent, giving avidity effects to the affinity that the Ty1 has. And this molecule is extremely potent. So now we get a 50% neutralization at concentration of, well, 12 nanograms per mil approximately. So that puts what we call now Ty1Fc into the range of some of the better antibodies that have been described this year for neutralizing the virus. And so we're kind of excited about this, and we hope that we can at some point go forward, and we're talking to some companies where we might be able to develop or put a version of this molecule into some sort of clinical trial in the future, we hope. So, and it's somewhat of a side project that we're just preparing for publication now. So this is the Ty1Fc molecule that I just talked about. We also made dimers by directly linking them, either with short linkers or long linkers. And we also made a tetramer where we stick it on this molecule that's called 4-arm PEG, which is, we're able to conjugate 4 nanobodies. So it becomes a tetravalent molecule. And we're able to do that entirely in vitro. And these are the products of those reactions run on gel. And then we perform the pseudovirus neutralization assay. And this is the data I'll just go through very quickly. So what we found was, remember, this molecule here was the very potent one I showed on the previous slide. And that molecule, together with the short dimer and the long dimer, all three of them sort of behave quite similarly. So still very potent, but the size of the molecule doesn't seem to matter, which is kind of interesting. But what's really exciting is that this tetramer is now much more potent than the dimer. So even this very potent Ty1Fc molecule, the 4-arm PEG tetramer, is now neutralizing in the picomolar range. So it's an extremely potent molecule. Problem is, though, of course, that the production of this is a bit more difficult than making the Ty1Fc molecule. But nevertheless, it shows the versatility of these molecules and how their modularity can be harnessed to make these kind of molecular tricks to make things more potent. And we also perform, this is for virology nerds who want to see actual plaque reduction titers. So these are the plaques of SARS-CoV-2 virus, where the virus has gone in and killed cells, and we stain so that the holes in the monolayer are then, plaques, so we can quantify the virus. And Ty1 neutralizes the production of the plaques. So as you would expect, it blocks infection, and so you don't get plaques. And the tetramer is also very potent at blocking, so not just the pseudovirus, but also the real SARS-CoV-2 virus. So that's kind of exciting. We're going to submit that as a small report very soon. But back to the main story, which is already published. We were lucky to have, so not just was Leo the expert on nanobodies and Ben the next-gen sequencing guy, but just across the corridor we have a cryo-electron microscopy group, or a structural biologist, Martin Hellberg, who was very interested in this the cryo-EM structure that was presented by the McClellan lab. And so as soon as we had the production of that complex working in the lab, we gave some of that protein to Martin and his colleague Rishikesh, who were able to solve, just like the McClellan lab did, the structure at pretty high resolution of the pre-fusion stabilized spike complex of SARS-CoV-2. And so they were working with that and perfecting it while we were cloning our nanobody. And then when we did have the Ty1 molecule available, we gave it to them, and they were able to very quickly solve the bound complex. So the complex of the viral spike in complex with the nanobody. And this is just a space filling model just to show that there's a couple of things I want to draw your attention to. Again, so the colors are different now, but again, this is the RBD and the up conformation. And what we see is that Ty1 binds at an epitope that largely overlaps, not entirely, but largely overlaps with the epitope that binds to the receptor. And so that kind of explains why the nanobody is so potently neutralizing and blocking infection. If the antibody is there, the protein is not able to bind to the receptor, quite simply. But what I also want to show you is that the Taiwan, that there's 2 other Taiwan molecules. So this is a trimer, and there is 3 molecules of Taiwan bound. So that the nanobody is able to also bind to the RBDs that are in the down conformation, which you might remember I told you is thought to be an immune evasion mechanism of the virus to hide the receptor binding domain until perhaps the receptor is in view. And conventional antibodies don't seem to be able to bind to those down conformations, but the nanobodies can, because of their small size and maybe because the CDRs are a little bit longer than those of conventional antibodies. So we were very excited about this. And here's shown in the side and the top view as well. So you see the nanobodies really do sit on the ACE2 interaction surface. If we go into a little bit more detail, so Martin and Rishikesh were able to solve the molecular details of the structure. And just to show again, Taiwan here overlapping with the ACE2 interaction surface. We can also show that, I thought this was interesting with something that Helen said about the CDR2 of the new antigen receptors in the sharks. We find that CDR1 and CDR3 are bound to the RBD, but CDR2 is not. It's taking up a position that's perhaps stabilizing the CDR1 interaction. So that was kind of interesting in that structure. And I also just want to point out that the steric clash that blocks the ACE2 interaction with the spike is conferred by the Taiwan bound at the RBD. But even if this molecule isn't here, the neighboring nanobody, Taiwan molecule bound at the neighboring RBD in the down conformation, is also conferring a steric hindrance to the ACE2 molecule as it comes in to be bound by the RBD. So this double steric clash, we believe, is a reason why Taiwan maybe is a little bit more potent as a neutralizing nanobody than some of the other nanobodies that we've shown and others have shown. When you have a binding in different conformations, it's not able to block at a lower concentration. So that was published we put it on bioRxiv. Leo claims that from the day we received the B cells from the animal until the day we put the manuscript on bioRxiv was 5 weeks. So this just shows that this work can be done extremely rapidly under pandemic circumstances. So bioRxiv allowed us to get that work out to the international audience. It then took a few more months for it to be published by conventional peer review process, but it's now out and in a very similar format to the bioRxiv paper. Just my last slide really is just to say that it took us a while to get the production of the spike complex working in the lab, but once we did, we sent it to our friends in pre-clinics research farm, and they immunized a second alpaca, and her name was Funny, this time a female, adult female alpaca, and Funny received 4 immunizations of the full spike complex. So, you know, expressing antigens in their native conformation is always a better strategy, and also the larger the proteins we found tend to be better antigens as well. And so for a combination of those reasons, we are now cloning out a panel of very interesting nanobodies from this animal, and her serum, by the way, you'll recognize this data perhaps from what I showed earlier. This is Tyson's neutralization activity in his serum. But Funny has many, many times more potent neutralizing activity. So there's a lot of very interesting nanobodies there. And we've already cloned a few that are very potently binding to the RBD, maybe more so than the Taiwan molecule. And some that we think are binding to other epitopes outside of the RBD, but are also blocking infection. So we'll be very interested to see how they're binding and if they tell us anything about the spike protein as well, or if they're maybe, you know, useful for diagnostic assays or perhaps even in potential therapies. So that's where we are with that follow-up project. And so it's been a very interesting year, as it has for many people. So I told you that Ben and another lab, Nila Cross and Hedestam, and I together applied for money from the EU, and we received funding to set up a consortium, which we called CORONAB, Coronavirus Neutralizing Antibody Consortium, developing antibodies in mice, in macaques, and also in alpacas. And so that funding really allowed us to do this work and in a dedicated manner. And we were able to recruit some people to help us at short notice. So Leo and Ben really are the main players in this project. They performed a mammoth task this year. Some other members of my lab and other labs are listed here. And yeah, I'll stop there. And the KI Development Office also gave us some funding. And yeah, so that's where we are. So as I said, I'm not really a nanobody expert, and I absolutely fascinated by the field, and we hope that we can go back to our old projects maybe sometime next year and show that these molecules are useful also as molecular tools to study virus replication, which was our original interest. So thanks for your attention, and I'll stop there.
Well, thank you very much, Gerald. This is an impressive work, and we are all very grateful for you for your research, right? And it's one more time we need to show society how important is research and giving funding, because yeah, it's really, really important. Thank you. Great talk. So we are going to pass now to the question and answer section. So I have here some questions for you guys. So I'll start. So for Gerald, since Taiwan is an excellent candidate as an intervention against COVID-19 or SARS-CoV-2, are these antiviral in clinical trials already? I guess you've said that you were waiting for. So what is missing? Like, are you waiting for the clinical trials to start? Or
no,
what,
I mean, yeah, what's missing? It's a good question. We just been we weren't really set up for that step. We had sort of prioritized everything else, getting to the point of having it, and then okay, what's next? And it's not something my lab or any of us in the consortium have experience with, but we have been contacted since the paper went out, by a number of companies, and we're talking to some about perhaps putting together some sort of clinical trial. In the meantime, you may be aware of Regeneron and Eli Lilly that have monoclonal antibodies, so conventional antibody therapies that have received emergency use authorization in the US. And we were surprised by, how should I put it, how high the doses they were giving to their patients and how little effect was actually. So, I think maybe I'm paranoid, but I kind of think that maybe some of the companies we were talking to lost interest when those data came out, because, they're giving gram doses to patients, and then only small numbers of the patients were showing significant reductions in hospitalization time. So it was a really marginal effect that was quite disappointing. Nevertheless, I think that so one of the things we're doing, a very early step, is we're talking to a company that might be able to make the nanobodies into some sort of nasal spray or inhaler, where maybe someone who's very recently been diagnosed with a positive diagnosis can then be given some inhaler to sort of try to reduce viral loads at that early time of infection. I think one of the reasons why the antibody therapies are not working very well is it just takes too long to diagnose and to treat, and the virus maybe has already starting to be cleared, but the immunopathology is still present, and the antibodies won't help against that.
Yeah, but could have been that.
Yeah, sorry.
Yeah, perhaps if the production is that easy, somehow could have been these nanobodies or antibodies administered before the infection, like as a prevention type of treatment for those people who are more at risk. Like,
yeah. Yeah, so I mean, the monomeric, the nanobody, when it's expressed on its own, can be produced in bacteria, and we can make, even a small academic lab like mine can make, milligram quantities quite easily, and then of course bigger companies could make them in huge doses. Once you put it on the FC, then it's required that we produce it in mammalian cells, and so that sort of slows down the process a little bit, but it might be offset by the higher potency. So yeah, we'll see how it goes. I'm sure there are many other labs producing nanobodies that have been published and around about the same time as ours, and still more coming out. So I'm sure someone will be much further in this process than we are. So I'm excited to see what comes out.
Well, I'm looking forward to seeing this in clinical trials soon. Helen, I wonder, why evolution placed nanobodies in such a different lineages, like camelids and nurse sharks, and how these unusual molecules might benefit these animals?
So I think, like everything, it's an evolutionary accident. So these things just happen, but then when they're found to be beneficial, they get kept on and they get used. I mean, we see that so often in the immune system, as if it's useful, we keep it. Sometimes even if it's not that useful, we keep it. So kind of evolutionary accident. In terms of then what they're used for, definitely it just gives a different mode of binding. So, both from my talk and from Gerald's talk, you see that these things are able to do jobs that conventional antibodies can't. So then those animals that have them actually have an advantage and can use them and generally keep them. Definitely from our work, we get really good responses against viruses. So one of my hypotheses is maybe, these things help target these epitopes on viruses that aren't usually seen by conventional antibodies, that viruses have evolved to hide or shield. And these little molecules can get into crevices or get between sugars that conventional antibodies can't, so give those animals an advantage against viruses.
And I wonder, how do sharks switch on their antibody manufacturing cells? Do we know much?
We are actually trying to figure that out. That's one of our big jobs. So, yeah, we're about 20 years behind the mouse and human field in terms of sharks. So we're doing a lot of work. The kind of the foundations of all this, like what are the B cells doing, where are they coming from, how are they activated, all the good stuff, figuring out memory and affinity maturation, and trying to figure out the mechanisms behind it at the minute. Mm-hmm. Keeps us busy.
Yeah, that's good. For Gerald, Taiwan binds the receptor binding domain of SARS-CoV-2 with high affinity, but what is the efficiency of this union? How efficient is the binding? You were saying before that there were other labs that produce also nanobodies, but with less efficiency. So,
well, I shouldn't say that. I mean, there are plenty out there that report high affinity binding as well. I'm not surprised. I don't think it'll turn out to be a difficult epitope to target with antibodies or with nanobodies. And so I don't think that we're been extremely lucky or extremely skilled or anything like that to find this one. It's just we report our one, and others are reporting also. I'm not sure I quite understand the question. So the affinity measurements were performed by biolayer interferometry and some biophysical assays that through collaborations that I'm not terribly familiar with, but the data suggests it's a quite high affinity binding. But for the reason why conventional antibodies are bivalent is because you kind of often rely on the avidity effects to get a strong binding, and we don't have that. So which is why the dimer and the tetramers perhaps are more strongly neutralizing, because there is a dissociation rate that's higher than we would have liked. So it's coming off, but when you have two of them on there, yeah, it stays on. Yeah.
Yeah, I guess because it will be really important, no, to have the unstable union when you administrate this, for example, in patients. So you really want to have a long lifespan of the antibody.
Yeah, and one thing that's really important is that it has a much higher affinity than the ACE2 molecule. So the receptor RBD ACE2 interaction is relatively low affinity for the reason that it doesn't need to be high affinity. It just needs to bind and then undergo the conformational change so that the virus can. So it hasn't evolved to be a high affinity interaction, and so as long as you have an antibody or a nanobody that has a higher affinity than that, that's the sort of main job you need to the main bar you need, the first bar you need to overcome.
Mm-hmm.
And I think most of the any of the antibodies that have been described now this year have that, because it's not that difficult to overcome.
Has this been tested in mice?
We're doing that now. It took us a long time we ordered in so ACE2 in mice is sufficiently divergent that the virus can't infect mice cells or mouse.
Mm-hmm. Okay.
So there was a human ACE2 transgenic mouse, but obviously the demand for breeding pairs was extremely high earlier this year. So we could get a few pairs, and we started to breed them, and we will be doing that too. Hamsters and ferrets are also a useful model. So they don't need to be transgenic. So the hamsters can be infected and they lose weight when they're infected, and you can use the weight loss as a measure for protection. And we're going to be doing some hamster challenge experiments as well.
Nice. This goes for Helen. Do animal-derived nanobodies bind to their targets better than the entirely lab-made alternatives? If so, why?
Usually. And that's because the animal kind of does the hard work for you. So it does the rearrangement and it does the selection. So, I've worked with non-immune libraries, with synthetic libraries, things like that. You get binders out of them, but they tend to be lower affinity and slightly less specific. Whereas, you kind of, and that can be improved. So you can always work on that, and you can mutate things and you can improve affinities, like in the lab. But, for me, it's easier if the animal does that bit for me and it does all the selection. We've had 500 million years to refine that within the animal. I let it do the work, and then we tend to get much higher specificity and affinity antibodies out. I think that's why most people still immunize. Even the llama system, I know there are synthetic libraries, but most people still immunize.
Mm-hmm. Mm-hmm. Yeah.
Can I ask about the temperature that you showed for this Ebola virus antibody? I mean, so this is extremely temperature stable,
mm-hmm.
which I was quite surprised by. For
mm-hmm.
What's the body temperature of the shark, and why did they?
Yeah, so I mean, on nurse sharks, they're actually around kind of 24, 28 degrees C. So it's actually cooler than human antibodies. But they have really high levels of salt and urea in their bloodstream. So most of their proteins are more stable than their human counterparts. So we've actually, our proteins have kind of relaxed more because we don't have that. But the other thing is just being single domain. They have quite a few disulfides holding things together. And what we've shown is it's like the camel antibodies. It's a combination of, they're more resistant to degradation and they're more resistant to denaturation, but also when they denature, they're able to refold back to their native conformation. They don't just crash out like conventional antibodies do. So it's kind of a dual thing that's going on.
That's really cool.
Mm-hmm. Yes, pretty amazing. This one is for Gerald. Gerald, I've read your paper, and then you mentioned that exchanging the hydrophobic residues in framework 2 may improve, you've shown the amino acid sequence. that the Tie-1 nanobody efficiency, so that may improve the efficiency of the, I guess, the binding. How this research is going? Are you doing anything of that at the moment?
We are not following on that. No, I don't.
Okay.
I don't remember why we would have written that. That's not.
Okay.
No, we did order some. So we did some mutagenesis of the protein. I don't remember if those residues that we referred to there are in that. But we did test a few mutations in the CDRs and in some of the backbone regions, and they didn't seem to make much difference. We weren't able to make a better nanobody than Tyson already did.
And another thing is that we don't know about the long-term effects of this SARS-CoV-2 virus in, for example, in the brain tissue. So do we know if can nanobodies pass the blood-brain barrier to treat diseases, for example, in the brain? I've heard that, I guess that for the size they can.
I'd have to point to Helen for that question. That's not.
Yeah.
If they're engineered in the right way, so there are some nice fusion proteins that hook up to transporters that can get across. There's a lot of work going on looking at brain targeting with these things. The idea being smaller, they should be able to get across.
Okay, thank you very much. And I think I'm going to ask the last question, and then I'm going to ask you some. We are going to start with a little discussion about the scientific system. So any of you, I think, can respond to this question. How long these nanobodies have been used in immunotherapies? And yeah, like if you think that these antibodies will be the future of synthetic antibodies.
Yeah. Well, I mean, one slide I often show, I didn't today, but they're, these are non-human, of course. So at least for the llama and alpaca. So there was last year one nanobody that has passed FDA approval for use in humans for human therapy. So it is possible that they can be humanized by just a few mutations in the protein to make them sort of less immunogenic and safe. So I do believe that they have a future in therapy. Absolutely, I think it's just starting. So we'll see where we can go with that. As I said, my lab's primary interest isn't in that direction, but we were caught up in the SARS-CoV-2 wave, and being able to produce this nanobody so quickly, we thought we have to try we should try to exploit it as much as we could. But our primary interest is using nanobodies to study the proteins that have been quite difficult to study. And we hope to continue to do that as well.
Well, thanks.
I think I totally agree with that. Going back to it's about the tools that are in our toolbox and picking the right one for the job. And sometimes, I think about, I teach med school, and we talk about how literally we use horse serum to prevent people dying of snake bite and things like that. It's like, it's not a great tool, but it's a tool. And sometimes, we can, things like immunogenicity, well, if it saves your life and keeps you alive, we can deal with immunogenicity later. So sometimes it's just about having those tools available and using the best one for the job at the time, and then fixing whatever problems come up later.
Yes. I think they are going to be an amazing, well, they are already an amazing tool, and they will be, and the amount of applications it's pretty impressive. So I'm going to start asking you, I'm going to ask you a couple of questions about what do you like the most about your work, and what are the biggest challenges that you face? And then what do you think we can do to improve these challenges that we face in science? So if we can start with Helen, for example, and then we can finish with Gerald.
Oh, Lord, what do I like?
To wrap up.
I like things like this, where I get to meet super exciting people that are doing really seriously cool research and learn all about it, and hopefully find ways to collaborate and do all this super fun and exciting and impactful science. I mean, I feel like that's a real privilege that we get. Frustrations, I mean, scientifically just my lack of tools, but that's because I work on a crazy non-model system. I guess sometimes as a female in science, coming up just against the fact that sometimes you don't get taken seriously, or sometimes people will, you know, I've been doing my job for 20 years, but people will try and tell me how to do it. And that gets really frustrating, is that sometimes you're not considered to have the same level of expertise as somebody that, you know, that's male and of an equivalent level. And that gets very frustrating from time to time. How we fight back against that? Literally to encourage more females in science and change the dynamic. I think that's all we can do. And the same, you know, with underrepresented populations and things like that, is we've really got to all be pushing for one another and pushing for more diversity and pushing for different points of view. And I think that will help us get around some of the issues that we're facing now.
Well, we have a great example recently with the Nobel Prize awarded to Doudna and Emmanuel, no? Jennifer.
But I would love to see the day where that's not a big deal. That's not reported in every single, I mean, it's great that it is reported in every single newspaper, but it shouldn't, you know, at this point in time, how many female scientists we've got, it shouldn't be newsworthy.
Yes, yeah, I agree. I totally agree. And you, Gerald?
Yeah, I mean, I totally agree. You know, yeah, one of the great things about being a scientist is you know, going to conferences, which we can't do anymore, but meeting people and collaborating. And when one has the time to sit and read papers, it doesn't come around very often. But when you sit back and read a paper that's really exciting, it's a great feeling, and one which I think I would like to have more often, especially if it, you know, gives you a new idea for your own research. Challenges are, you know, for me, it's the boring one of just scrambling around for funding all the time, and it's just been a constant struggle that just shows no sign of going away. We were lucky to get the funding we got this year, but of course it runs out, and then we're back looking again. So that's a kind of a boring answer, but yeah, administrative burdens as well can be a bit, you know, would be nice to be able to do less admin, but maybe that's being greedy because someone has to do it. And people often complain about teaching burden or teaching obligations, but I love teaching and I do a lot of it. And I'd love to be able to do more, but I'm finding that my teaching is now coming with even more admin as well. And so I find myself being an educational administrator rather than a teacher. And so that's frustrating, because I've always had the desire to have both in my career, to be a scientist and have a little lab and do cool things and be a teacher. And that's just really difficult to be able to do both, because there are no positions available for teachers who want to do research, or researchers who want to teach. It's usually, you know, you have to be world class at one or the other, and if you are, then you don't have time to do anything else. So that trying to balance that, you know, these opposing forces in my career is a challenge, but it's so far it's been working okay.
Hopefully the governments realize that we need more funding and we need to facilitate.
Yeah, well, I mean, I hope so. I think that virology as a subject is going to be maybe more respected in the future after the COVID. Well, the damage is calculated for this pandemic. In the end, is going to be astronomical. So preparedness for the next one, which is undoubtedly there will, of course, be more viruses emerging. The question is, how much damage will we allow them to wreak?
Yeah. Well, thank you very much for your time and for your talks. I really enjoyed, and this feeling that you were mentioning of reading a paper and getting excited, I had it with both of your papers, well, with different papers of your research. So I'm very grateful. Thank you very much. And with this, I'm going to finish this webinar. This webinar is going to be, it has been recorded, so we will share it in the social media platforms. Thank you very much. It's been a pleasure to meet you. Bye. Have a good afternoon and have a good day, and I'm going to have a good night.
Yeah.
Thank you.
Thank you. Bye.